Okay, Science 30, so this is a continuation. We're going to now talk about pedigrees. So pedigrees are basically a family tree. And so they tell us how a trait has been passed on in a family and who has it and who doesn't. So some symbols that we have to understand here before we get started. Circles are females, squares are men. If it's shaded in, they are affected by that trait. And to be affected means that you just have the trait. You could have a disease and be affected by that trait that's genetic disease, or you could have blue eyes. And if you have blue eyes, then you'd be shaded in because you're affected by the blue eye genes. And if you don't have blue eyes, you would be not shaded in because you would not be affected by the blue eye gene. You'd have some other eye color. Lines between a male and a female mean sexual mating. Lines come vertically up and down from two parents to an offspring mean that they have had a kid. So this line here means mating. This line here means that they have had a kid. Um, Offspring are always listed in birth order. So from left to right, you get um, who was born first, who was born second. So from these two kids here in generation number one, from grandparents one and two here basically, they had three kids, an oldest son, a middle child who was a daughter, and a third child that was a son. That son married a woman, and they had one kid who was affected by whatever trait we're looking at. Roman numerals are going to dictate what generation you're talking about. So generation one, generation two, generation three. Okay, let's get started. So um, we can basically label a pedigree multiple ways. So first we want to label the generation. So this is clearly generation one, Roman numeral one there on the left, generation number two, generation number three. Let's label each individual. So this is person one of generation one, person two of generation two, persons one, two, three, four, five, six, seven in birth order of the kids for generation two, and generation three labeled. We could say some stuff here, right? We could say that these two had one, two, three, four kids. One, two, three of them were girls. Only one of them was boys. This one was affected by a trait. This one was not affected by a trait. This one was affected by a trait. This one was not affected by a trait. This one didn't marry anybody. The other three kids did marry, and then they had these kids. Okay, so let's answer some questions here. How many children did generation one, and generation one, two have? So generation one, person one, and generation one, person two, these two. How many kids do they have? Well, they had one, two, three, four kids. What was their sex? Well, like we already said, three girls, one boy. Which one was the oldest? Well, person two, number one, this daughter over here. Okay. Assuming that having dimples is a recessive trait and no dimples is a dominant trait, write the genotypes for all the individuals in the pedigree. So now we gotta label the pedigree. So we gotta write a legend. So we'll use capital letter D for no dimples because they said that was the dominant trait. And then having dimples is going to be a recessive trait. So we'll give that a lowercase letter. So now let's label everybody here with a genotype based off of if they're shaded or not. Well, this person is not shaded, so they must have one dominant no dimples gene. This person is shaded in, so they clearly have dimples. And the way you can have dimples, being a recessive trait, is if you have two lowercase letters, so little d, homozygous recessive. You can label everybody in generation two in the same way. If you want to pause it here and try this out on your own, see if you can figure out the labels of these guys in generation three. Okay, here's generation three, all labeled. So quite simple, right? Once you have a legend, you're just figuring out who gets what letters, who gets what genotype based off of if they're shaded or not. Okay. Continuing in this example then, what is the chance the next child of individuals 2, 1 and 2, 2 will have dimples? So 2, person 1, 2, person 2, to these two. There's their genotype. Mom's homozygous recessive, dad's heterozygous. What's the chance that their next kid will have dimples? So you got to do a Punnett square. You got to do a cross. So there's your cross, big D, cross with a little d, little d. Do your Punnett square. Dad was little d, Dad here is little d, big D, little d. Mom was little d, braid, top down, left in, top down, left in, top down, left in, top down, left in. Fill in your Punnett square. 50% of them are going to have dimples. 50% of them are not going to have dimples. So 50% are not going to have dimples. The next child will have a 50% chance of having dimples. What is the chance that the next child of person 2-4 and 2-5 will be a boy with dimples? Now, they're not only asking what's the chance that they're going to be having dimples, but what's also the chance of them having dimples and being a boy. And we're looking at two different parents now, 2-4 and 2-5. So generation 2, person 4 and 5 are right here. Both of them have the homozygous recessive genotype, little d for both of them. So we got to do a Punnett square. I'll move my little face over here. So there's my one parent, there's the other parent. We're going to fill in the Punnett square, top down, left in for everything. Boom, they're all the same. Well, the question was, what's the probability of having dimples first? Well, 100%. 100% of them are going to have dimples. What's the chance of being a boy? Well, you either are a girl or a boy. So you're either going to have a 50% chance of being a boy or a 50% chance of being a girl. So to answer questions like this, where it's asking what's the chance the next kid is going to be a boy with dimples, you are going to basically multiply the two probabilities together. There's a 100% chance from these two parents of having dimples, and there's only a 50% chance of being a boy, because the other 50% chance is you're a girl. If you multiply the two together, so 1.0 times 0.5, or 100% times 50%, you get 50%. So there's a 50% chance of being a boy and having dimples, according to this cross. A Testcross is something that you do if you're not sure if somebody who expresses a trait, a dominant trait, is big T, or big T, little t. They're going to have a dominant trait no matter what, but are they purebred? Do they have homozygous dominant big T, or are they homozygous, or are they heterozygous? Big T, little t. Both cases are going to show the trait, but one's purebred because one doesn't have the little t gene in it and one does. And so if you're not sure, you do a Testcross. This is important for people that are like breeding cows or dogs or something. You want a purebred. You don't want to be breeding something that isn't pure even though it looks pure. So you're going to do a Testcross to figure it out. So a Testcross is used to determine the genotype of an individual who expresses the dominant trait. So what is a Testcross? Testcross is used to perform to perform a Testcross, we will cross the unknown plant with a homozygous recessive little t plant, or whatever. It could be a dog, cat, sheep, cow, doesn't matter. To do a Testcross, you always cross your unknown dominant with a heterozygous recessive individual every time. So you have two options. When you perform a Testcross, you are either going to, have a homozygous dominant individual, or you're going to have a heterozygous individual. If you have a homozygous dominant individual, all of the offspring are going to be the same color. The alternative is if you have a heterozygous individual, only half of them are going to have the same trait, and the other half of them are going to have the opposite trait, or the recessive trait. So let's do an example. It'll make more sense. So two options. Option number one. Let's say you cross a unknown tall plant. You don't know if it's big T, or big T, little t. And you have to do it with a short heterozygous little t, because that's the way Testcrosses are done. You do your cross. There's your Testcross little t, crossed with your unknown tall. And let's say when you did that cross, you found that all of the offspring were tall. All of them had the dominant genotype. So there was no short offspring. In that case, the unknown is likely going to be big T, because that's the only way you could produce all tall offspring, is if there was a big T to bring in, 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 because these little t's were providing all the little t's for all these options. Option number two, however, is if you do this cross with a Testcross, and now 50% of them are tall, and the other 50% are short. And so if two are tall and two are short, then you have some short offspring. If you have some short offspring, then the unknowns are likely going to be big T, little t, because there's some little t's getting in there. So if this was big T and this was a little t, there's now some little t's getting in and making your kids different than what you'd think. So this way, you can figure out, okay, well, I'm not going to use this individual because they're not purebred. I'm going to go back and use the other individual that made all the same offspring when I did the test cross because they're more likely purebred. Okay, in the next video, we're going to talk about sex linkage and genes.